7:15. So, good evening. I'm Judy Noodleman, a Westminster resident since 2024, and I'm also the founder of Women in Neuroscience. And what you see up here actually are pictures from our interns across the different years. This is our 9th summer of summer internships. So if you look at this, right here, it's year number 1. And then you can see excerpts from every year.
No,
No,
not
grab
even.
the microphone or grab the microphone and come on out.
All right, so let's grab the microphone and come on out. Thank you.
Keep it close,
So I founded Women in
Oh, Judy.
Neuroscience in
closer, right up there.
I'm telling them this and I'm forgetting. I founded Women in Neuroscience in 2018 to honor my husband Harvey's career in neuroscience and my career as an early woman in STEM with a 30-year career with IBM.
Get the mic. Oh, somebody's on the mic.
Not fast enough.
So WIN's mission is to accelerate progress in solving the complex challenges of neurodegenerative disease.
Good.
We do this by increasing diversity of thought and experiences in the neurosciences as we empower students from under-resourced and underserved communities. WIN does this by enabling these students to spend 9 summer weeks in a lab, gaining experience important to their future. By helping these interns also, many economically challenged, so they can take advantage of this opportunity, we provide a $5,000 stipend. And we also do this by staying connected and assisting and mentoring the interns after the internship is over. A great example of that is our 2025 intern, Anna, who was a presenter here at Westminster in our August 2025 presentation. Through connections we facilitated, Anna is now a lab manager at New York's Icahn School of Medicine with John Crary, a world-renowned neuropathologist. Anna's future plans include graduate school in neuroscience. Hi. All right, So, to ensure we increase diversity of thought and experiences to further solve the challenges of brain disease, we incorporate diversity as one of the selection criteria in the application process. And you can see through this chart, in our 9 summers, 89% of our interns are women of color and 49% are first generation. In 2026, we received our first Westminster Resident Association grant. With matching funds from 25 Westminster residents, we provided the $5,000 stipend for Irene Ducky, for WIN's first Westminster Scholar. Irene. We thank the association and the residents for these donations. In the beginning of the summer, many of us got to meet Irene and had lunch with her, and tonight, all of you will get to meet Irene and learn more about her. We're excited also that she has been invited to continue her work in the lab during this next school year. And this is super exciting since she is going to be applying to medical school, and that should have a major impact. So tonight, in addition to hearing from Irene, you'll hear remarks from our program director, Valeria Marquez Cardenas. Valeria was a 2024 and 2025 intern, and after she graduated in May from UT Austin in Neuroscience with honors, she joined us as our program director. Yay, Valeria. You'll also get snapshots of the work of many of our summer interns who are here in the rows. And then after they've all presented, you'll get to ask questions. So they're going to talk about a lot of details. If you have some questions, I suggest you jot them down, because by the time you get to number 2 or number 3, you may not remember what you wanted to ask number 1. And then you'll get details about tomorrow evening's research symposium and impact assembly at Dell Medical School. There's still time to register, and the Westminster bus will be there to take you. So with that, please join me in welcoming Valeria.
Thank you, Judy, so much, and thank you everyone for being here. I don't know if any of you were actually here, but I actually was here a couple of months ago trying to recruit participants for a study, and I was recruiting participants that have suffered a stroke. That have suffered a stroke. So that's actually the reason why I got my departmental honors. So it was all of you that made that possible. So thank you very much for that. Additionally, I just want to say that all of our interns are very, very excited to be here. They have practiced very hard, and what you're about to see is a culmination of their work. But before you see that, I just want to briefly talk about programs and what we actually do at WIN. So our flagship program is our internships. Our internships provide a $5,000 stipend for our interns so that they can focus on their work and reduce the financial burden that is living in Austin and in Austin's economy. Throughout the internship, they have a variety of things, but the thing that we're proudest of is that they have 35 hours of lab time per week. In this time, they're allowed to basically learn what it means to be a scientist. They get one-on-one mentorship with a faculty host, which is a very skilled researcher in a very specific area of neuroscience. So their skills that they're learning are very specific to that topic, which again, you'll see today. Additionally, they have weekly required activities. Those are primarily the speaker series, which is neurologists and neuroscientists come and speak to them. They also learn a lot about professional development. They have people coming in and reading their resumes, their cover letters. We actually have one of them here, Gail Kaplan. And overall, they just get more well-rounded, as we know, and as many of you might actually know if you happen to interact with a scientist. An issue that we have a lot of the time is that scientists typically don't have those soft skills. So that's what we want to do during those weekly activities. We want to make sure that we not only nurture their scientific endeavors, but that we're also nurturing their personalities and their personhood, make sure that they are still connected to what it means to be a human being. Additionally, we also have socials and events. We actually went a couple of weeks ago to a museum and we had a nice time. We explored the Blanton Museum. If you've ever been, I highly recommend it. And then the culmination of their work is actually what you're gonna have a sneak peek of today, which is the research symposium. All of our interns that are here are gonna give you a brief snapshot of what they've been preparing. And if you're really interested, I highly encourage you to come tomorrow. As Judy said, there's gonna be a bus that is going to take you directly to the event. So we hope to see you there. So now I would like to welcome Irene to give her presentation. Irene, can you tell us a little bit about yourself?
Good evening. My name is Irene Dochi, and this summer I was given the opportunity to work in the Loop lab under Dr. Jared Lewis Peacock with the guidance of my mentors, Edward and Kayla. I am a rising senior at UT Austin, and just like Judy said, I will be applying for medical school this upcoming school year. This summer, I worked on a project that looks at the effects of suppressing information in working memory. Let me move to my poster, I'm so sorry. Okay. So before we get into the poster, I want to outline the idea of working memory. It is a system that temporarily holds information in mind so that we can use it for ongoing tasks and behaviors. For example, it is the reason why you can read the beginning of a sentence and then read the end of it and understand the full sentence for comprehension. You can hold it in mind to truthfully understand what it's saying. Does that make sense to everybody?
Yeah.
Great. So now let's go into an imaginative headspace. Do we all use Facebook here? Is that something that we all use? No? Okay, maybe like, okay, we watch the news perhaps, right? Perfect. Okay, cool. So imagine that you're in your home watching the news, okay? You see a news article or a story about a neglected puppy, if you were to see it on the news, I don't know. Okay, you see something about a neglected puppy, so sad, you move on. The next thing you see on the news is a family that lost their house in a fire. You know, you feel for them, maybe you're thinking of ways you can help. Nonetheless, you keep watching the news, and the next thing you come across is someone sharing their story about how they're suffering from a terminal illness. You keep watching the news, you see one more news article about a mother who lost her young child. Then you decide this is too much negativity for you. So you turn off the TV and you go to your desk and begin to work on a personal project. 20 minutes later though, while you're working on your project, you start to think about that neglected puppy that you saw, or maybe the house fire that you saw. You know that this information is not relevant for the work you're doing currently, but it still sneaks up on you, so you try to remove it from mind. Well, previous studies have shown that you actually can remove this information from mind, and there are different strategies to do so. The most effective strategy is something called suppression. Suppression is what it sounds like. It's actually pushing something out of mind. It's not replacing your head of all thought, or maybe like clearing your mind of all thought or replacing that with something else. It's actively trying to push it out. It's almost like trying to hold back a cough during a presentation. So, going back to the news articles that you saw all of those sad news articles. Now you're in a headspace of negativity. Just like at some other point in life, you can be in a headspace of positivity. The issue is, studying this kind of emotional information, has only looked at emotion in isolation. We haven't really looked at the context behind it. And as you and I both know, when we feel things in the real world, it's often embedded in some sort of context. So, this pilot study I worked on this summer attempts to explore this idea of emotional context and suppression. This leads me to my research question. Does a negative emotional context hinder suppression? We predict that yes, a negative emotional context will hinder suppression. It has been found that negative information and negative images are more remembered, so that means they're harder to suppress in general. So if you take this idea on top of the idea of an emotional context, a negative emotional context, we predict that it should be even harder to suppress this type of information. Now I'm going to walk through what participants saw in this study. But before I do that, I want to give some background and keywords. So to emulate this idea of emotional context, the 4 images they saw were presented in a particular sequence. Let's go back to the idea of seeing the news, right? You're watching the news. Let's say first you saw a news article of, I don't know, maybe a 4th of July celebration, right? That's something positive, right? Then the next thing you saw was the news article about the neglected puppy. But then after that, if maybe someone's marriage proposal was on the news for some reason, I don't know, that's what you saw next. And then the last thing you saw was the house fire. That would be the interleaved context. It's positive, negative, positive, then negative. It could be negative, positive, but either way, 2 positive images, 2 negative images. The grouped context would be you watching the news, seeing something positive, and then you saw all of the negative things afterwards. In the grouped context, there is either a positive or a negative majority. So, 4 images appeared sequentially, following one of the contexts that we just talked about. After all of the 4 images appeared, we saw a cue. If the cue was a green circle, participants were instructed to maintain this image in mind. The image that appeared in that position, try to hold it in mind. If it was a red X, they would have to practice the process of suppression. Then, after the cue went away, a black frame appeared around one of the positions. And this indicates what image appeared in this position. So then on the right side of the screen, participants saw 2 images, an old image and a new image. The old image is the image that actually appeared in that position, and the new image is an image they had not seen before, but had a similar valence, whether it was positive or negative, as the original image. Then participants made a decision. So if everyone is following, I want to move now to my results. Okay, we'll start there. So graph 1 shows reaction time on the y-axis and operation on the x-axis, maintain versus suppress. For all of the graphs on the screen, green represents positive images and red represents negative images. We found a significant effect of operation in reaction time, but not in accuracy. So essentially this is saying that participants significantly responded slower to images they were told to suppress than those they were told to maintain. And this is cool because it replicated previous findings within this field that yes, indeed, suppression reduces the accessibility of these images. Moving on to graph 2, it is showing how well participants performed according to the context the images were in and their valence, whether they're positive or negative. So that's what we see on the x-axis, and on the y-axis we're looking at accuracy. There was also a significant effect here. This means how well participants identified negative and positive images depended on which context they were in. Looking particularly at the interleaved context, participants were more accurate at identifying negative images and positive images, which we knew that. You know, negative things tend to stick with us more, right? These results suggest that the mere existence of an emotional context affects how differently participants respond to negative and positive images. Graphs 3 and 4 look at suppression across valence and across context. For these graphs, I would predict that images would be harder to suppress within the grouped condition than within the interleaved, but across both contexts, negative images would be harder to suppress. So looking at graph 3, I'm gonna go down to the middle. On the y-axis, we have the change in reaction time found by reaction time for maintain minus the reaction time for suppress. Positive values indicate a faster reaction time for suppression than maintenance, and negative reaction times indicate a slower reaction time for suppression than maintenance. And that is what we see here. All of the values are negative. But I didn't find anything significant. Keep in mind, this was only a pilot study with only 10 participants. And those of us in science know that we need way more than 10 participants, right? So this kind of just shows proof of concept. Visually, what we see here is that there is a longer reaction time for the negative images in the grouped condition than the interleaved condition. But again, nothing was significant, so all we can say now is that in the grouped and interleaved context, suppression reaction time was similar across both valences. Now let's look at graph number 4. Again, suppression across context and valence. On the y-axis though, we're looking at the change in accuracy, found by the accuracy for suppression minus the accuracy for maintain. Values above 0 indicate that there was a greater accuracy in recalling suppressed images than maintained images. And values below 0 indicate the opposite. Let's go back to our story of watching the news to really make this make sense, okay? So let's say you were tested on the news article of the neglected puppy, right? You were told to maintain it, then you were tested on it. If you were able to accurately and correctly say that, yes, this is the news article of the puppy that I saw, that means you successfully maintained the image. And now let's say you were tested on it, but then you were told to suppress the image instead. This would mean that you unsuccessfully suppressed the image if you were able to correctly say that this is the image that I saw. So here, there was no significant finding, but visually we see that in the group context, bars were below 0, so suppression was more effective. And in the interleave context, they were closer to 0, a little bit above 0. So suppression was less accurate here. Just to quickly go over my discussion, essentially what we see here is that suppression reduced the accessibility of information, but did not differ reliably across context and valence. And so emotional context may shape memory performance without particularly disrupting suppression itself. But again, a pilot study, so the next steps would be to collect a greater sample size to see if we find significant results. I'd like to thank the Lupe Lab, Dr. Jerry Lewis Peacock, Wynn for this opportunity, and most especially all the Westminster residents that gave so that a girl like me could be in this position. I really appreciate it. Thank you.
Thank you so much, Irene. Another round of applause, please. That was awesome. Now, briefly, for a few minutes, we just want to open up the floor for any questions that you might have for Irene. Just raise your hand and I'll hand over the mic.
What was the result of your stroke study?
Oh, we can talk about that later. For now, let's stick to Irene's questions, but I'll talk to you.
Irene, was it just because that's what the lab is working on, or was this a particular interest of yours?
Well, this is what the lab focuses on, working memory. This particular project was more interesting to me because there are things like PTSD and anxiety that have been shown to be caused by an imbalance in working memory. You know, we take in so much information every day, and people with those clinical conditions have a harder time managing them. And as someone who wants to go into medicine and I want to become a psychiatrist, I felt like this was a good gateway into kind of understanding the idea of emotion and working memory.
Could you explain the valence part? We can't hear you without. Could you just talk about what the valence part means?
Yeah, valence is whether the image was positive or negative. So there was a
Okay.
data, a big bank of images. Some were pretty bad. I know some of the interns did the study. Some of them were pretty, and so they saw positive images or negative images. Are there any more questions?
One more question.
Speak of it if you would, your path to where you are today.
Yeah. Oh gosh, this is an interesting question. So my family is from Ghana in West Africa. My parents and my older sister were born there, and they came here and had me and my 3 other siblings. I grew up in Rhode Island, in Providence, a kind of a lower income community, I would say. Had the opportunity to move here to Texas for college, and so I go to UT. I'm going to be a senior. Yes, go Longhorns. Yeah, and so I think I chose neuroscience because I had a friend in middle school who died from brain cancer, and so I always wanted to do something with the brain for sure. I thought I wanted to be a neurologist for the longest time, but I think different experiences in college and like my own personal mental health and just like podcasts, I listened to a lot of podcasts about mental health and like psychiatric conditions, kind of pushed me in the direction of wanting to help particularly incarcerated individuals and how their mental health may have affected them to be where they are today, and how we can sort of better them. You don't want to forget about anybody in the community. So
Thank you so much, hearing you answer those questions beautifully. Thank you. Now, for the rest of the night, what you'll be seeing are basically snapshots of what you'll be seeing tomorrow. So each presentation will be 3 minutes. We will cap it at the 3-minute mark. In case you see people wrapping up kind of suddenly, that's what's happening. But yeah, before we do that, I just want to briefly introduce you to some pictures of the interns that you know who's talking. We have Catherine, Chelsea. Janelle unfortunately was not able to make it today, so she won't be here, but we have her picture in her honor. Jessica, Adriana. We have Elisa, Lilian. Estrella was also not able to make it today, unfortunately, but again, in her honor, she's here. And then Alex. So let's start with Kathy. Come on up. And just like Irene, please tell us your name, major, and the research that you're doing, and the lab that you're at.
Good evening, everybody. My name is Catherine. I go to Center College, which is in the middle of Kentucky, but I am currently a rising junior and a neuroscience major. Can you guys hear me?
Yes.
All right. So over the summer, I worked in Dr. Dunsmore's lab in the Dell Medical School in the Department of Psychiatry, and my project focused on using Pavlovian fear conditioning to retroactively strengthen near subliminal images. So think of when you go through an emotional event, whether that be winning a competition or going through an accident, something that's gonna stick with you. So those memories are usually very intense. But what also happens is the strengthening of the weaker memories that occur directly before and directly after. So suddenly you remember the shoes you were wearing, or what the weather was like, or just a meal you ate in the morning. And so my project focused on understanding how weak a memory can be before your brain is no longer retroactively enhancing that memory. So can images or can a memory be subliminal? So can it not even really tap into your conscious memory, but just flash for a few milliseconds and then get strengthened once you go through an emotional event? So my project focused on flashing pictures of different semantic categories. So we use animals and tools for varying lengths. So for 12 milliseconds, 100 milliseconds, and 2000 milliseconds. And that 12 millisecond gets really close to the subliminal line, which is what we're interested in. And so I wanted to explore if this effect will carry on to subliminal memories, so very weak memories. So the shocked category, so to increase that, to elicit that emotion, we shocked people, and that was the fear conditioning part. So we shocked them to either an image of an animal or images of tools, and we had many, many different images of animals and tools. So just to keep it balanced, some people would get shocked to animals, some would get shocked to tools. And so for the first round, so the preconditioning phase before they were shocked, they were shown the images for varying amounts of time, 12 milliseconds, 2000, 100, in varying different patterns. And then they were shocked to one category, so just tools or just animals. And then 24 hours later, they were shown a bunch of pictures, some that were new, some that were old, and asked to rate whether they're old or new in a level of confidence between 1 and 4. So 4 for very confident, 1 for old, I didn't see that, I'm very confident that I didn't see that. So these are our results, and so unfortunately there wasn't a significant effect of the subliminal images. So the effect does not carry on onto very weak memories, so they have to be at least consciously registered before they're able to be enhanced retroactively. So if you just walk by, if you just For example, if you go through an accident and you just have like a red bike that flashed forward or just flashed by you, you wouldn't really remember that. You'd have to at least see that bike for a few seconds, recognize it, and then you'd be able to remember that afterwards. However, once we split the data into people who got shocked by animals and people who got shocked by tools, so this is people who got shocked by animals in the blue, people who got shocked by tools in the light blue, we did see a significant difference in the people who were shocked only by animals. Anyway, thank you so much. I'm gonna have to wrap this up. I wanna thank my lab and all of my interns. Chelsea?
Hello everybody, hi everybody, I'm Chelsea Guzman, and I'm currently a rising sophomore at Southwestern University in Georgetown. I'm a kinesiology major and a neuroscience minor, and it's been awesome. This summer I worked in Dr. Lippard's lab at Dell Medical School in the psychiatry department and had a lovely time. My research this summer focused on sleep characteristics and related alcohol expectancies in young adults with bipolar disorder as I completed a placebo-controlled alcohol administration study. So sorry. Okay. So this research is important because majority of it has been done on adults. So I am part of the limited research that's been done on the population of young adults. So I'll be focusing on ages 21 through 26 with a mean of 23 years old. And oftentimes, bipolar disorder and alcohol abuse disorder co-occur, but the factors are unknown. Also, sleep disruptions may be a risk factor for alcohol use disorder throughout its effects on cognition. The 3 study aims that I focused on throughout my research are 1, examining sleep differences in young adults with bipolar disorder, investigating clinical factors associated with sleep disruptions in bipolar disorder, and exploring relationships between past week sleep changes and placebo responses to alcohol. Moving on to my methods, I looked at a sample size of 104, 48 being healthy controlled young adults, and 56 being young adults with bipolar disorder. I measured their sleep using the Hamilton Depression Rating Scale, focusing on their sleep changes and their sleep characteristics, specifically their wake-up time change, their bedtime time change, and their sleep duration time change in comparison to the past week, and their usual sleep patterns. And I focused on the prevalence of disturbing dreams that they had throughout that. The beverage administration study consisted of 1 placebo day and 1 alcohol administration study, and they completed this in a simulated bar lab, which I'm happy to tell you guys more about later. And whenever the participants reached peak breath alcohol concentration at 0.08, they completed an array of self-reporting surveys, and I focused on looking at their valence and arousal responses, specifically the anxiolytic effects and the sedative-woozy effects throughout their placebo session. Moving on to my results, as you can see in the first graph, the bipolar group and the healthy control group had limited differences whenever looking at their sleep duration change, so they both had similar patterns. However, whenever looking at the prevalence of disturbing dreams, the bipolar participants reported more than double the amount of disturbing dreams than our healthy controls. Now moving on to the clinical characteristics. I focused on looking at cannabis use disorder. And as you can see, the participants with a positive cannabis use disorder experienced more significant sleep duration change in comparison to participants without a cannabis use disorder. I also looked at ADHD, anxiety, and alcohol use disorder comorbidities with bipolar, but saw no significant differences. Cannabis use disorder was the only one. Moreover, moving on to how wake-up time change and anxiolytic effects correlated, in black, it represents the healthy controls, so the later they woke up in comparison to normal sleep patterns, the less significant anxiolytic effects they experienced throughout the placebo session. However, in the orange line representing my bipolar group, the later they woke up in comparison to normal sleep patterns, the more they felt the anxiolytic effects throughout the placebo session. Moving on to the research the results that I found most surprising was that affected both my healthy controls and my bipolar group. They both responded experiencing disturbing dreams, however, and they experienced more sedative woozy effects. However, that wasn't the case whenever it came to feeling rested. It didn't have the same effects, looking at the same characteristic of sedative boosting. Thank you so much for listening to my presentation. I'm happy to answer questions later.
Okay, hello everybody. My name is Jessica Apt. I'm a rising third-year neuroscience major at UT Austin. And this summer I got to work with Tom Alston, who's new at the Department of Neuroscience. So overall, the goal of my project this summer was, to understand how people use self-control to base their decisions on relevant information for goals while ignoring irrelevant information. To kind of make a little more sense, we all know my favorite grocery store, H-E-B. So let's pretend that we are there right now, and we're all on a diet. We're walking through, grabbing our groceries for the week, and we get to the aisle where they have all the chips, the snacks, and whatnot. If someone had high self-control, they would be able to walk through this aisle, recognize that they have the goal of staying on this diet, and ignore the irresistible crunch of those chips around them, therefore successfully focusing on their diet. On the other hand, someone with low self-control would not be able to do this. They would grab that bag of Lay's, throw it in their cart, and continue on, failing to focus on their diet. We were able to make a task that measures this in a lab. So participants were able to do a task where they looked at a pair of colored shapes. Each color had a value, and each shape had a value. In each trial, they were shown a word on a screen, either the word color or the word shape. When they saw the word color, it meant that they should focus on just the color values and ignore the shape values. The opposite for when they saw the word shape. A key thing about the study is that they were able to make multiple decisions before locking in their answer. Remarkably, people who had conscious knowledge of what the goal was did not do so good at this, and as the irrelevant information increased, they had a lower accuracy rate. A key finding from my study this summer was this graph where each dot represents one participant. On the x-axis, you have the initial error rate, which means it's like grabbing the chips and putting them in their bag. On the y-axis, you have the correction rate, meaning they could put it back on the shelf. 3 groups came out of it. We have the purple group, the high control group, meaning they rarely grabbed the chips and put them in their basket, but if they did, they almost always put them back on the shelf. The yellow group is the group that almost always got the chips, but never put them back. The blue group was so-so in both metrics. And what this means is we've successfully made a task that can measure different control levels in individuals. We want to tie this in the future to psychiatric symptom disorders. So we know some things like ADHD, OCD, etc. can hijack your behavior with the irrelevant information. And our goal is to connect the 2 findings together. So yeah, I really thank my lab for everything they've taught me and all the friends I've made this summer. And I'm happy to answer questions later.
Hello, I'm Adriana Netzold. I'm an upcoming junior at UT Austin. I was in Dr. Laura Fonkin's lab. Studying circadian rhythms, which is like our day and night cycles, with microglia, which is our immune cells in our brain. So, okay, so to start off, my project was focused on neurodevelopmental disorders. Which is autism, schizophrenia, some that we have heard of. But usually these disorders, the origins of these disorders and why they happen are unknown. But currently research has seen that it's an interaction of many complex factors. Currently we're focusing on what's called a 2-hit model. there's a genetic predisposition and an environmental factor that react together and increase your susceptibility to developing these disorders. So, for example, we're studying circadian rhythms. If we were to have a genetic predisposition to knock out all those rhythms in our cells, we would be more likely in developing neurodevelopmental disorders. Additionally, let's say we get inflammation a lot of the times. Females will have inflammation when they're giving birth, and that could also lead to neurodevelopmental disorders. And both of the examples I just gave interact through microglia, our immune cells in our brain, which survey your brain environment and are able to fight against inflammation. And so if both of those interact, maybe we can do something with these microglia to fight against this vulnerability. And specifically, we decided that if we put receptors into these microglia and activated them, we were able to boost those rhythms to fight against that inflammation. So I did a pilot study using a drug and these receptors in microglia to see if we could boost the rhythms in the first place. So this is just a simple little method, but we can focus primarily on how did we even see what was going on. We did immunohistochemistry, which is just staining these brain slices and being able to see the activity. And so, the drug works. Whenever we give DCZ to these animals, there's more activation. Even without quantifying it, you can see that there's a lot more dots when given DCZ. And in our first immunohistochemistry, we see that those receptors I was talking about are in green. The CFOS is cellular activation. CD11B shows this like purple color is the microglia, those immune cells, right? And so females, we see that the dreads are the receptors are activated, but they're not on the microglia. Kind of strange. And then in males, we see that the dreads are on the microglia, but they're not activated. So maybe our staining was a little weird. So we decided to try a different activator, activation stain. And if we take a look, we can see that once again, the GFP are those receptors I was talking about. But this time, the red is gonna be our microglias. And if we look at females, the dreads, or the GFP, is not on the red. And in males, they are. So these dreads are localizing to places that aren't just the microglia. So if we were to give this drug, it would activate more than just what we wanted. So why could that be? Particularly, we think it's either a sex-based difference or an inheritance-based difference. Meaning that if it's sex-based, males and females just differ in where these receptors go. But if it's an inheritance difference, then maybe they inherited it in a different way that caused them to be localized differently. But yeah, thank you.
Hello everyone, my name is Elisa Robledo. I'm a rising junior at UT Austin and I'm studying neuroscience. So this summer I had the privilege of being in Dr. Arredondo's lab at the Child Development and Learning lab where we work with toddlers, so that was really fun. For my research project, we focused on functional connectivity, specifically resting state functional connectivity in the brain of toddlers, which represents when different brain areas are working together. So when I refer to connectivity, it doesn't mean functional connectivity, like you would connect two wires together. It's functional, meaning they're working together. So it is important to accurately measure resting state functional connectivity in toddlers for the early identification of atypical connectivity that would be associated with disorders such as ADHD and autism spectrum disorder. And very little is known about the types of stimulus that are used while studying resting state connectivity in toddlers, so that is what my project this summer looked at. And if you guys have any toddlers in the family, they love Bluey, the cartoon. I don't know if you guys have heard about it, but that was one of the stimulus we used in our study. And along with Bluey, we used a bubbles stimulus, which consisted of a research assistant blowing bubbles in front of the kid, as you guys can kind of tell over here. And for the study, we recruited 41 toddlers ranging from 24 to 38 months. And 25 of the toddlers watched the Bluey episode, whereas 16 watched the bubbles stimulus. And we used FNIRS, which is known as functional near-infrared spectroscopy. You can see over here that the headband we used. And just to give you like a really quick example, if you look at your Apple Watch, turn it over and measure your heart rate, turn your Apple Watch over, you're gonna see a green light. It's very similar to what we use for FNIRS, but FNIRS uses a red light. And that measures the levels of oxygenated hemoglobin in the brain. I can show you later, Judy, for sure. So yeah, we used that headband, and then we ran some statistical analysis. We ran individual correlations for each of the participants, and then ran correlations comparing the two groups. So Bluey versus a bubble stimulus. And I didn't mention this, but they wore the headband for 7 minutes while being engaged in the stimulus. For fun graphs over here, this displays the connectivity while watching Bluey and Bubbles. And I'm going to specifically focus on this one right here. Excuse me. This one right here at the bottom. That represents the difference in connectivity between the Bluey and the Bubble stimulus. So what we saw was there was much greater connectivity while watching the Bluey episode than while watching Bubbles. And that kind of references a greater cognitive load while children watch the Bluey episode than while they engage the Bubbles. And this means that watching a cartoon episode that is a story-based stimulus is way more cognitively taxing than watching Bubbles and engaging in a more naturalistic stimulus. And why does this matter? Well, when we're focusing on resting state connectivity, we want to look at how the brains of kids are wired at rest. So this might suggest that a cartoon-like stimulus, such as Bluey, may not be as effective for measuring that signal and may actually confound our results. So yeah, I would like to thank all my WIN interns, the WIN leadership, and the Child's Lab for their support. Thank you guys.
Hello everyone, my name is Lilian Bolaños. I'm an incoming junior at the University of Texas at Austin, and I am also a neuroscience major. This summer I was able to work in Dr. Sisler's lab, or Neurotap lab, in the psychiatry department. So today I will be going over a little bit about my project and going over the goal of my project. It was to study whether childhood trauma, in addition to trauma that occurred later in life, was associated with circulating anandamide levels and hippocampal gray matter volumes. So for a brief background, childhood trauma is a big risk factor for the development of PTSD, and it has been associated with long-lasting alterations in brain regions involving learning, memory, and emotional regulation. The endocannabinoid system plays an important role in regulating stress, fear extinction, and emotional processing as well. Anandamide, one of the primary endocannabinoids, has been implicated in PTSD in the past. It may also represent a biological mechanism through which childhood trauma can influence brain structure. So moving on to the methods of my study, participants were gathered from previously enrolled Neurotap studies, and their clinical, biomarker, and neuroimaging data was collected. Blood samples were used to quantify circulating anandamide levels, and structural MRI data was used to obtain hippocampal gray matter volumes. All data analyses were done in RStudio, and participants were categorized into 3 separate trauma exposure groups. One group consisted of participants with only adult trauma, the other consisted of adults with childhood trauma only, and then the last group consisted of participants with childhood plus adult trauma.
And here's a small graph with my participant sample sizes. And just to make some quick points, participants ranged in ages from 21 to 50 years old, and the sample was predominantly female, making up over 85% of all participants across all analyses. Moving on to my results, Figure 1 shows the biggest finding that I found in this project, which was that there was a significant negative association found between lifetime trauma exposure and left hippocampal gray matter volume. But this was only specific to the childhood plus adult trauma exposure group. Figure 2 shows that there was also no significant finding between lifetime trauma exposure and circulating anandamide levels within each specific trauma timing group. Figure 3 shows that there was also no significant finding within the centered anandamide levels and hippocampal gray matter volumes within each trauma timing group. So to quickly summarize my project, while circulating anandamide concentrations were not associated with trauma exposure or hippocampal volume, analyses did identify that there was a significant negative association between cumulative trauma and the left hippocampal gray matter among participants with both childhood and adult trauma exposure. And that was basically a summary of my project, so I'll be glad to answer any questions. Thank you.
Good evening, everyone. My name is Alexandra, and I am a rising sophomore at Texas A&M University, majoring in genetics. Today, I'll be talking about a little bit of my project with the Bastilli Lab, resulting in the successful replication of associations between impulsivity traits and brain structure in adolescents. We primarily focused on impulsivity as it is known to have consequential effects on the mental, behavioral, and personal relationships of children starting from childhood. Now, for our study, we specifically looked into the data from a 10-year longitudinal study that is known as the Adolescent Cognitive Development Brain. Sorry, guys, can you hear me well?
Yes.
Oh, yes. There we go. So we mostly worked with the data from a 10-year longitudinal study known as the Adolescent Brain Cognitive Development Study, funded by the National Institute of Health to analyze brain and health development of children. Now, this study mostly consisted of 12,000 participants starting from the ages of 9 to 10 years old across 21 different sites in the United States. The main scale that we worked with was the UPPS-P scale, which categorized impulsivity into 5 different categories. It consisted into negative urgency, positive urgency, lack of perseverance, lack of reasoning, and sensation seeking. Now, this scale was mostly applied to individuals through different questions, and they primarily worked to understand if there was a relationship between impulsivity traits and gray or white matter. Now, gray matter is the outside of the brain, while the white matter is the inside of the brain. And it was primarily looking at the association between impulsivity traits to structure. Now, the primarily methods used consisted of about 12,000 participants from the most updated database that had quality MRI scans as well as quality answers to the UPPS scale used. Now, we primarily conducted multiple female linear mixed effects regression analysis for each of these impulsivity traits in its relation to gray and white matter throughout cortical thickness, surface area, volume, including FA and MD. Now, our most significant results consisted of that there is a positive relationship between impulsivity traits with gray matter regions. For positive urgency, we mostly saw it in a negative association, meaning as the impulsivity traits of positive urgency increased, the relationship to structure decreased. Now, for lack of planning, there was a positive association. For negative urgency, there was a negative association. And for white matter tracks, for 4 out of the 5 impulsivity traits that we looked into, 4 of them had a negative association with brain structure. Now, we were able to successfully replicate 35% of the analysis for DMR analysis compared to the original study that was looked into in 2020. Now, for 50% of the DMR analysis were able to be replicated, and we would like to take the study a bit further in following years to look into the association between impulsivity traits and brain structure across the different ages, starting from 9 ending to 19 years old, and then also possibly looking at the genetic associations between impulsivity traits. Thank you very much for your time.
Would all of the interns come on stage, and we're going to spend a few minutes answering any questions you all have. There will be a microphone circulating, and Valeria as well. Valeria? So any questions that you have for any of us, including Valeria or myself,
I couldn't resist. How many of you used AI to evaluate your research once it was finished? None of you?
Does anybody want to answer?
No? May I suggest that after you finish it, ask it?
I don't want to be a nerd about AI. So the way that it actually works is it takes everything on the internet and then it gives you an answer based on what's already out there. So if you ask it to evaluate what's already out there, it won't give you anything new. It'll tell you what it already knows, and then it'll throw some words in to make it sound new. So if any of us used it, we wouldn't really have real research.
Good answer.
Yeah.
I want to confirm that your study of the impulsivity was all based on data that was already out there.
Yes, so the data we used is from a 10-year longitudinal study called the Adolescent Brain Cognitive Development Study that has already been going on and is still currently in progress across different sites in the United States. So yes, it's previously collected data used to replicate studies that's been used for the same data, but to ensure that they're replicable and accurate, to ensure that that the information we're proving to the public is accurate.
Thank you for that answer, Alex.
any other questions?
Do we have any other questions? Yes.
I do. Chelsea, how did you know when somebody had a bad dream?
We had a couple of surveys that each participant completes first whenever they're screened to see if they're eligible to participate in the study. But we use another survey called a combined clinical. So part of my study also includes an MRI scan before they complete either the placebo session or the alcohol session, but I didn't look at that specific data. But on the day of their MRI scans, they complete the combined clinical is what it's called, and they answer a set of questions of what their sleep is like, their sleep patterns, what they feel like, and yeah, they answered pretty honestly. And so yeah, it was, the way that I differentiated was just a plain yes or no. So if somebody said sometimes, then I marked that as a yes, they had a disturbing dream, and yeah.
Thank you.
So we're all, go ahead, Hank, you'll be
Just
the
one quick
last one.
question. Number one, you're all so incredibly impressive. I mean, amazing. And I just want to ask a general question. Before you started this program, did you have any idea that you would be accomplishing what you have? Anybody care to answer?
Just pass it down.
to be honest, I did not imagine I would have the opportunity to give a presentation such as this to such a big crowd. I am very fortunate to have received this internship, and I definitely did learn a lot, including as to where I want to take my career further in the future, and yeah.
No, I had no idea what the, like, and what of this would be like. I've learned that research is like, it's a roller coaster. I've had days in the lab where I've actually cried. There are days where I leave and I'm so happy. There are days where it's like, ugh, nothing is working, you know, like, it's actually just like, again, a roller coaster, and I'm so grateful to have actually experienced this. Like, I designed my study from top to bottom, did every single step that it takes to, you know, analyze data and all of this fun stuff, and it was a lot, but like, I would have never experienced this anywhere else. I'm really grateful for this opportunity.
Yeah, on a similar note, I think being able to like look at our posters and know that we're the ones who can take ownership of all the things we've done, along with like all the people who've poured into us in our labs, like our mentors and such. Like, I've made so many mistakes and they've all like helped in some way. And so to be able to do those over the summer, to like make mistakes and learn from those and then eventually produce something like my poster is really cool.
And I had a very similar experience. I really had no idea what research looked like. This was my first time ever working in a lab, and I had an amazing and positive experience, and I'm actually going to continue working in my lab throughout the school year, which I'm super excited about. So I would definitely say it shaped what my future is going to look like. Now I'm steering more towards a research route for my future, and it's something that I don't think I would have had the opportunity to do if it wasn't for WIN.
I had a similar experience to Chelsea. This was my first time ever being in the lab since the school I go to is so small. It's actually a college, not a university, so we don't really have any big labs. So I had a wonderful experience. I learned how to make the poster, how to design a study, how to run human participants, and I'm really grateful for the opportunity that I had.
Same for me. I agree with every other intern, and especially all the help that we've gotten. I don't think any of this would have been possible by myself, especially knowing that I tried to do things by myself, and this poster wouldn't have been possible if I didn't have my PI, my mentors, this whole program. And I'm really grateful for the support, and it has really shaped who I am professionally and in my future and in so many different ways. I'm gonna have to agree with each and every one of the interns. Being at UT, it's such a huge school, it's really competitive to get into research labs nowadays. So I feel very lucky to be in the lab I am right now, especially since I can relate to my grad students and like my PI on a personal level with their experiences and what they've gone through. And I'm just very privileged to be there, and I'm so lucky to have all these awesome interns. Like, I wanted to cry when I was listening to them talk, they're so amazing. As well as like all the leadership at WIN. So I don't think it would have been possible really, so I'm just really happy to be here.
Funny enough, I came to WIN very confident that I was going to go into like a graduate school program with research after, but I had never been in a lab. And so then I got here and I joined one, and I think one thing that was reinforced was I knew it was something I wanted to do, but my lab brought me bowling and we tried different foods from all over the world all together, and I think that was my favorite part of learning the community that I will one day make when I become a research host, and I'm very excited for
Thank you to our interns. We're going to conclude in 1 second. If you didn't get the handouts at the back, there's 2 flyers there. One will show you our website and a little synopsis, so you can use the QR code to get into what those are. And then the second is a poster about tomorrow night's research symposium. So if you would like to learn more, which you will tomorrow night, by listening to some of our alumni who have been on this road and are at the major much further along, please join us. You can still sign up, and I checked with Ruth, you can get on the bus as long as you sign up by the morning. Thank you all. stay around in case any of you have questions.